I want to thank the organizers for allowing me to speak about my project today. So I'm going to tell you a little bit about the human reference work that we've done at the McDonnell Genome Institute. So at the McDonnell Genome Institute, we've been a part of the Genome Reference Consortium since the beginning. The Genome Reference Consortium was formed after the human reference was complete in 2003. A few years after that, it was realized that there were issues, there were problems with the reference that needed to be addressed, and even just gap filling and normal issues that you would potentially have with a genome. So the GRC is made up of Sanger Institute and the McDonnell Genome Institute that have been doing the wet lab work that was needed to fill some of these gaps and solve some of the regions. And then the EBI and the NCBI have been the bioinformatics portion of this consortium. And then more recently, ZFIN has also joined, and they work on zebrafish alone. I should mention that the consortium was formed initially for human and mouse, and zebrafish is also now included. At McDonnell Genome Institute, though, we primarily just work on human. So as we started to work on the reference, we realized more and more that it really is a work in progress. There are a lot of updates that have been made to the reference and continue to be made throughout the years. So the current reference is GRCH38, but we know that it's not fully representative of some regions of the genome or even some individuals or ancestries. The reference is currently comprised of DNA from many individual human alleles. And we know that allelic diversity and structural variation can present major challenges when assembling a representative diploid genome. In more recent years, newer technologies and methods, as well as resources, have allowed for substantial improvements in the reference, but we still realize that additional high-quality reference sequences are needed to represent the full range of genetic diversity. So 5 to 6 years ago at MGI, we started a project funded by NHGRI to sequence additional human genomes. And this is a map of some of the ones that we started with. The original plan consisted of sequencing 5 diploid individuals, and these are samples that were part of the 1,000 Genomes project, so they already had some minimal amount of sequence, but we were gonna create references from them. So like I mentioned, we planned to do 5, we have already sequenced 30. So as technologies have increased and have gotten cheaper, we've been able to do more than we originally planned. So our original sequencing plan was to do 60x of PacBio long-read data, and then do a de novo assembly with that data, and scaffold with either 10x, dovetail, or BioNano, and then potentially to align to the reference. In the very beginning, we had a few back libraries created for some of these samples, and actually would sequence the backs in regions that are very hard to assemble on a whole genome basis. That ended up being too costly, so we ended up with only about 5 of the total samples having back libraries generated and sequenced from. But then the goal of the project was to end up with a fully sequenced reference genome of that individual sample. So far, we have 21 of those genomes in GenBank submitted, so they're freely available for download. And I should also mention that most of our raw data is either already submitted in the SRA or in the process of being submitted, so you can play with it if you would like. The majority of these genomes are submitted as a contig level assembly. So those are draft assemblies. They're not ordered oriented contigs. And then the first three up there are ones that we actually did take to the chromosome level, but we decided for the rest to kind of go another way, and you'll understand a little bit more as we go through this. So this was our assembly generation, QC, and submission pipeline. We would generate a Falcon assembly of the PacBio data, and then one of the things that we found was that there were a lot of little parameters that you could tweak in Falcon to get a better assembly. So we would end up doing that and decide on what the best assembly was. From there, we would run Quiver with the PacBio reads to do error correction, and then also Pylon, which uses Illumina data to error correct the consensus. From there, we would use bionano alignments to the whole genome assembly to identify potential chimeric contigs, and generally, we would have 50 to 60 chimeric contigs that we would want to break before we would submit, and then we would submit these to GenBank. So here's a graph showing the assembly, the contig in 50s as well as the scaffold in 50s. So in blue are the contig in 50s from the Falcon assembly, and this is just a portion of those samples. One of the things that you'll notice towards the end here, I don't think this works. There's a few samples towards the end of the graph that have a very short contig in 50 for the PacBio assembly, and those were ones that were generated with the Sequel 1 when we first got it. The majority of this data was actually sequenced on the RS2, but then after we had the Sequel for a while and we're running it, the contig in 50 went back up, so we were able to get the read length figured out, and everything was kind of back to normal. So the red bars here in the graph are the bionano, the sapphire maps that we created from the bionano DLE maps enzyme, and the contig in 50 for those are generally around the 50 to 60 megabase mark. And then in green is the hybrid scaffolding, so scaffolding the bionano and the PacBio assembly together. And we would end up with somewhere in the 60 to 70 megabase as far as a scaffold in 50, which this is really getting close to chromosome arm length. So, you know, they're fairly complete, or we thought they were. So CYP2D6 was mentioned earlier. CYP2D6 is a variable region in individuals, and it is a drug metabolism gene, so it's a very important gene. One of the things that we started to realize as we would look at these assemblies is we would have the bionano data as well as the initial Falcon assembly, and frequently there were things that we would find in the bionano data because it actually separated the alleles better than what the Falcon assembly would do. So in this instance, the green bar at the top is the reference, and then the two blue bars are the two alleles in 12878. And one of the things that you'll notice, the area marked in red, is an insertion in allele 1 of 12878, but also there's an insertion in allele 2, which is different than the first allele. So here is a heterozygous individual for this important gene, where our assembly, the way it was, will only show one allele. So, you know, we've generated all this data, and yet then we're losing part of it. So this really pointed out the need to have a diploid separated assembly. So we work with Jason Chin when he was at PacBio, and we were working with Falcon and then Falcon Unzip. So Falcon, as it forms the initial assembly graph in places of the genome where there's little to no discrepancies between the two alleles, it will form a single path, and then in regions where there are discrepancies, it forms these little bubbles. And then Falcon Unzip, as it goes through, it will actually cleave or basically separate off the second portion, and you'll end up with one pathway that goes all the way through. That's the primary assembly. And then the shorter contigs end up going into a haplotig FASTA sequence as well. So in the very beginning of us looking at Falcon Unzip, it really didn't, it basically did not, it wasn't as contiguous in the haplotig bucket. It was a lot of really, really short sequences, and it didn't contain much sequence. So it wasn't very effective in the very beginning. So more recently, obviously, these things have improved with sequence improvements. So we have now been working with our Sequel II for a while, and with the release of the Sequel II, PacBio also released the CCS data. The CCS data was always around, but it wasn't something that we used very often. Right now, though, the improvements that they've made to the enzymes and the loading efficiency has really helped with the data from the CCS sequencing. So in this slide, I'm showing, in case you're not aware of PacBio, the contiguous long read sequencing, or CLR sequencing, is basically where we would have a really large insert into the SMRT Bell, and the enzyme would primarily only get around the insert once. So you would end up with really long reads, but they're not very accurate because the polymerase has only been through the read once. And in the CCS data, the insert is shorter, and the enzyme, as it goes through the circle, begins the displacement mode, and once it gets to that, it goes a lot faster. So you end up with many copies of the same sequence, and from that, the processing makes a consensus of that data. So in this diagram, you'll see the little red dots here, and those are random errors. And in the HiFi read, because the errors are random, when you actually put all the reads together, you end up with a high confidence read. So one of the more recent data sets that we generated with the HiFi data has been the 19240 genome. It's a Reuben genome that we sequenced very early in the project with the RS2. We wanted to go back and see how it compared with the HiFi data. So this is a fairly new data that was off of our instrument. PacBio agreed to actually assemble it for us, and was able to turn around the assembly in a matter of days. So here are some of the read plots and stats. This first one in blue is the raw read length, and you'll see that the reads are pretty long. You know, there's 100 kb there, or even higher, where the raw reads, so that shows how long the enzyme has been on the molecule. And then once it's processed into HiFi data, the read length is closer to about 20 kb. And in fact, the mean read length for these 4 SMRT cells worth of data was 18 kb. And then the last graph shows the HiFi read quality. And one of the benefits to the HiFi data, as I mentioned, is the high read quality. We actually have reads that have a quality of Q40, but the median of the read quality is Q30, which is really good. So in this, this is an IgE view where the reads were aligned to the reference. So these top 2 panels are just the reads themselves aligned to the reference. And you'll notice how well that those reads map and how consistent they are. Also along the bottom here, I wish the pointer worked. So along the very bottom, the last 2 tracks there are the actual assembly consensus. And you'll notice how well the assembly, the 2 alleles match up to all the individual reads. And there's a fair amount of, in this region in particular, there were a fair amount of homozygous as well as heterozygous SNPs, and you can see how well phased they are. The one towards the left is to your right, is a 500 base pair deletion in the one allele compared to the other. So PacBio did a coverage titration assembly for us where they assembled each of the, we generated 4 cells worth of data, and they assembled 1, 2, 3, and 4 total cells so that we could see how the stats changed along the, as we added more data. So you'll notice in these graphs that the plots, they really jump from 1 cell's worth of data to 2. But then when you move from 3 to 4 cells, they kind of even out. And the same thing is true for the number of contigs. When you have 1 cell, there is a fair amount of contigs, but then when you move to 2, 3, or 4, the contig numbers are pretty flat. So this tells us that at some point we might not need 4 cells worth of data, but right now we're continuing on that track. So then the next step that they did for us was actually to run Falcon Unzip. So they ran Falcon Unzip for the 4 cells worth of data and the 3 cells. And the thing to note here is that the quality is very high for both the primary and the haplotype assembly. What I had mentioned before was that the haplotype assemblies in the very early days of Falcon Unzip was very short and that didn't contain much sequence. So in this case, with the 4 cells worth of data, the one thing that you notice is that the primary assembly is 2.95 gigs, and the haplotype assembly is 87% of that. So 87% of the assembly also has a corresponding haplotype in the haplotype FASTA. It is pretty fragmented. The haplotype assembly is pretty fragmented, but the primary assembly is a pretty high contig in 50, so it's very contiguous. And then if you look over to the other side, you'll notice that the 3-cell assembly is very similar. So you're not losing much by not generating that extra cell's worth of data. So basically, in summary, why would we switch to the CCS data? Basically, the CCS reads are consensus reads, so they do have a much higher quality than the CLR reads. There's no beginning error correction step when you start to assemble the CCS data, as opposed to what you need to do for the CLR reads. Less coverage is needed, so 30 to 40x is basically where we're at right now for assembling CCS data. PacBio actually recommends 30, and we were previously generating over 60x for the CLR data. And the assembly time is much quicker. In the early days of generating the CLR data and doing the assemblies, it could take us weeks, if not even months, to get an assembly done. And now this data was actually turned around for us in a matter of days. So what's next? What's next on our plate, as well as a lot of other people, is this Human Pangenome Reference Consortium project. So, we know that a lot of sequences are needed to fully represent all ethnicities, and this project will start to address that. The goals of the project, we are going to start new sample collections so that we can actually ensure that all diversity is actually included. The first samples that we're sequencing are from Coriell, but with Coriell we're very limited on diversity. We plan to produce 350 high quality reference genomes, so that's 350 samples. So in reality, we would like to have 700 assemblies. We plan to produce finished telomere to telomere genomes. There will be an assembly evaluation pipeline to ensure accuracy and completeness. And we plan to develop a pangenome reference, so that will likely look a little different than the current reference. And then we'll need to develop tools to utilize that reference. Then we plan to continue to annotate and improve the reference the way we've been doing for GRCH38. So with that, I'll end the talk, and basically these are a lot of the people involved in the new pangenome project, as well as some of the old data that I generated. So we're out of time, but okay, thanks.